All right, good evening. My name is Hunter, and this is Content Review for Principles of Biology students 211. Week 8 topics. We're looking at the chromosomal basis of inheritance, building a little bit more on what we learned about last week. We're going to be doing a lot of Punnett squares. Sit tight. We're going to be looking a little bit more closely at some of those other inheritance patterns that we mentioned briefly, but didn't take too deep a dive into. So we're going to be treating this in terms of experiments. I really like these because they're kind of like it's just logic-based problems, and it's fairly simple to set up a visual that allows you to really understand what's the math, visually understand the math that's happening here. So I'm working on a slightly different program today because my preferred one is being really sad. Precise highlighting. Okay, so we're gonna do our best. An organism that has the genotype big A, big B, big C is crossed with an organism that has the genotype big A, big B, big C, little c. Now remember that capital letters denote dominance in a trait and lowercase letters denote recessiveness uh, for the same trait. So assume all the genes are on separate sets of chromosomes, that they are not linked, so there's no crossing over happened. We've mentioned this a couple of times, but we haven't really looked at what that means in the context of these Punnett squares. So right now, dealing with Mendelian genetics, there's no crossing over happening. The chromosomes are not linked. So we're going to get into that a little bit later in the worksheet. So what is the probability that any of the offspring will have the genotype just homozygous dominant across the board? Hint, to get to the answer, consider the 6 possible types of autosomal crosses. So autosomal meaning not sex-linked. Basically, this is just prompting us to build Punnett squares for these two organisms, for each of these traits, and go from there. So let's start with, we'll do our, I'm prepping these 3 Punnett squares simultaneously. We're going to do each of these separately. We've got our heterozygote across the top and our homozygote. Oh, excuse me. Yes, on the vertical. Excuse me. So we've got, yes. So our second organism is only homozygous dominant for that A gene. So that's all that's really changing here. I'm going to pick a different color just to keep it coherent. So we've got, yeah, this cross happening here. You can go all the way across, but we can kind of stop right here because the ones we care about is this homozygous dominant for all traits, right? So it occurs 2 in 4 times, so 1/2, 50% of the times for the A gene, 1 in 4. I'll fill this in so you can kind of see. 1 in 4 for the B gene, and 1 in 4 for the C gene. So to get our probability, we simply multiply those across. I'm multiplying the denominators in my head, so 2 times 4 is 8, times 4 is 32, gives you a 1 in 32 chance of big A, big A, big B, big B, big C, big C. And again, these are the genotypes that we're concerned with. Cool. Okay. Make sure you can read that. What is the probability that any of the offspring will have the genotype big A, big B, little c, little c? So slightly different outcome here. And we could just look at the same Punnett squares and highlight the desired ones if we want to do this visually. Come on now. Um, so big A, 50% of those. Big B, 50% of those. Little c, so 1 in 4. So we've got, this time, my stylus is not behaving. Come on now. Oh, don't quit on me now. Hmm. Don't make me type this, please. All right, stand by. I'm gonna make you look at my face while I problem solve this. I'm sorry. Okay, it appears we're back. So 50% of the A's, 50% on the B's, and 1 in 4 on the C's. 4 and 4 is 16. Make it a little bit clearer that that's a small c. Okay, so a 1 in 16 chance. Continuing on. Same cross. Oh, different cross. Consider the cross A, big B, big C, d, big E, times big B, little c, big D, big E. What is the probability that any of the offspring will have the following genotype? So, in looking at this, very briefly, we can't rule anything out yet. It would be really easy to rule something out, like if they were asking if there was a possibility of a dominant gene that wasn't present um, in any of the crosses. So let's go ahead and start. Let's go ahead and start building these, and hope that our hope that our screen doesn't crash. So we've got A, B, little c. Let me make that space a little bit bigger. Little c, little c, big C, little c, big D, little d, little d. We can already say with this D cross that 100% of those are going to be heterozygous. There's only one outcome there. And then we have, again, I'm crowding myself a little bit. Big E, little e, big E, little e. Do our crosses. So this one, because they're all going to be big D, little d, I can save myself a bunch of work just by doing that. Come on now. Okay. And then we just go component by component. Big A, little a. Big B's, big C, little c, big D, little d, and big E's. And then do our math. We've got 1/2 times 1/2 times 1/2 times 1. 4 out of 4 is 1 times 1 and 4. So 4, 8. 8 times 4 is 1 and 32. Oh, hopefully this is making sense so far. What is the probability that any offspring will have the genotype big A, big A, big B, big B, big C, big C, big D, little e? So I kind of forecasted this, right? There is 0% chance. We don't even have to do the math because anything times 0 equals 0, right? And just looking at these Punnett scores, we know that there's no chance of a big D, or a big C, because we don't have that dominant gene occurring twice. There's a 0% chance. I'm not going to belabor that point because I think this should be fairly intuitive, and it's fairly well illustrated just by doing the Punnett squares. Um, but if you're a stochastic thinker and you like the numbers, um, you can actually go and write those probabilities out and do the math that way if you like. All right, so next prompt. In fruit flies, Drosophila melanogaster, uh, really common experimental organism. It's very famous. The most common eye color is red. So red is going to be our dominant. Well, we don't know if it's our dominant one yet, but we can assume because it's the most common. A mutation or allele of the gene for eye color produces white eyes. The gene is located on the X chromosome. So what is the probability that a heterozygous red-eyed female fruit fly mated with a white-eyed male will produce any white-eyed offspring? So we have to set this one up a little bit differently. Because this gene is located on the X chromosome, this is a sex-linked gene. It is not autosomal. Because it's X-linked, we're going to have two different possibilities for that gene. Um, we're going to say that W would correspond to the dominant gene. So that would be our capital letter. And W plus corresponds to our recessive gene. And so here we have kind of a key for how we're talking about this. So this W right here is for the red, the dominant, and this W plus is our white mutant. Okay, so this notation is helpful because we're talking about a mutation in the gene and not a different. It is a different gene because it's a mutation that it can be passed down. So it you could speak of it in those terms, but it is the same gene varied. I don't know if that actually helps, but it makes sense to me to use a symbol this notation rather than dominant versus recessive. Okay, so what is the probability that a heterozygous means that we have a heterozygous female, so two X chromosomes, one with the dominant red eye gene and one with the mutant white eye gene, with a male with white eyes. Because this gene is linked to the X chromosome and males only have one of them, that gene will be expressed, right? That genotype will be expressed into that phenotype, and then we can make our cross. Let's keep it, I kind of crowded myself here, but that's okay. Start with X and X makes female, X and Y makes male. And remember that for these males, these male offspring, the X chromosome always comes from the parent female, right? So one of those males will have the dominant red eye gene, that's this one on the left, and the other one will have the mutant white eye gene. So here we have another heterozygote, and here we have a homozygote female. And because it's just kind of hard to see, I'm going to highlight where the mutant genes appear. There. Okay. So what is the probability that a heterozygous red-eyed female fruit fly mated with a white-eyed male will produce any white-eyed offspring? The probability is fairly high, right? These are good odds. But notice here, it's heterozygous recessive. So that gene is not going to get expressed. That particular female is going to have the dominant red eyes. So 1/2 chance of white-eyed, wow, eyed offspring. Cool. What is the probability that the mating in part A will produce any white-eyed females? Because this is a recessive trait, both chromosomes have to be carrying that sex-linked gene, and so our chance here is 1 in 4. The question is asking the probability that any of the offspring um, will be white-eyed and female. So the other way that you could do this is by simply multiplying those ratios together. So we know that 50%, yeah, I'm going to erase this answer so you can see this again. We know that 50% of all offspring will be female, and we know that 50% of all offspring will express that mutant gene. Therefore, the probability that the mating pair will produce a white-eyed female is 1 in 4. So that's how you would do the math without doing the Punnett square, if you were just given those raw numbers. What is the probability that this mating will produce any white-eyed males? Same story. I don't think I need to rehash this just because we can see it visually there, and um, and we've just done this math. It looks exactly the same. The probability of an XY pairing is 50% or 1/2, and the probability of that mutant gene being expressed is 1/2 equals 1 in 4. The difference here is the reasoning. The reason is because you only need one of those mutant genes to show up, and it just has to show up on the X chromosome. Okay, excuse me. Okay, a heterozygous, brown-eyed human female who is a carrier of color blindness marries a blue-eyed male who is not colorblind. Colorblindness is a sex-linked trait. Colorblindness is a sex-linked trait. Assume that eye color is an autosomal trait and that brown is dominant over blue. What is the probability that any of the offspring produced have the following traits? Okay. So let's look first at, we're going to be doing two different Punnett squares, I think. We're going to be doing one for eye color and one for color blindness. So let's look at the female. Goodness. I'm going to do this over on the left margin. So our female has to have two X chromosomes and is a carrier. So carrier would mean we have one normal gene for color vision and one mutant gene, not mutant gene, one gene for color blindness. Okay. They also, um, they're heterozygous for blue eyes. So that means they have the dominant gene for brown eyes and the recessive gene for blue eyes. And this is our female. Our male in this case has to have XY. And because they are not colorblind, is that right? Blue-eyed male who is not colorblind. So the blue-eyed male must therefore have homozygous recessive for blue eyes and for that gene to be expressed. And they just have the standard color vision. Because if the colorblind gene appeared on the X chromosome, it would be expressed. So, what is the probability that any of the offspring produced will have the following traits? Brown eyes. Well, let's do our Punnett square for brown eyes. We've got big B, little b, little b, little b. Looks like 50%. For blue eyes, also looks like 50%. For color blindness. Come on now. Stylus is having a hard time. So here we've got a carrier. Let's make sure you can see that. Top right of this Punnett square, we've got a carrier. Here we have a male who's not colorblind. And here we have one male who is colorblind. So color blindness would be 1 in 4. Colorblind males is also, oh, hang on. Probability of a male is 1/2. Probability of color blindness is 1 in 4. Is this right? Nope, it is 1 in 4. Because there's the probability of, yeah, sorry about that. Leading you astray. The probability of a colorblind male is still 1 in 4. Yeah, the probability of being colorblind and male is 1 in 4. Hmm. Yes. Okay. Brown-eyed, colorblind males. This is where it gets a little bit more tricky. So the probability of being brown-eyed is 1/2. The probability of being colorblind is 1 in 4. And so the probability of producing a brown-eyed, colorblind male is 1 in 8. I have questions about D. The probability of, well, okay. Moving on. um, what is the probability of producing blue-eyed, colorblind females? Similarly, 1/2. There is no probability of a colorblind female based on this pairing. Come on. I know it's not my stylus. I know that it is my app here that is causing problems. Okay. So there's a 0% chance of producing a blue-eyed colorblind female. What is the probability that any of the males will be colorblind? So this, okay, they do address this. This is asking a slightly different question. This question asks only about the males and not about all of the offspring. So if we look at all of the offspring, we find that a quarter will be colorblind males, but what is the probability that any of the males will be colorblind is simply 1/2. Why do males show sex-linked traits more often than females? Simply put, males have only one X chromosome, and therefore, come on, gene is always expressed and comes from or is inherited from parent female. Okay. And by contrast, this is important. By contrast, female offspring have two X chromosomes. Therefore, both must carry recessive gene or recessive allele to be expressed. Now we're on to solving problems when the genetics are unknown. So this is working backwards, right? And this is much more like, I think, what you would see in an experimental setup. Um, for example, with a wild population that you want to understand the genetics of. So an understanding of Mendelian genetics allows us to determine the theoretical probabilities associated with normal transmission of autosomal and sex-linked alleles during reproduction. This understanding provides us with strategies for solving genetics problems. In real-life situations, geneticists use these strategies to determine the genetics behind specific phenotypic traits in organisms. They do this by conducting controlled crosses of experimental organisms, or by analyzing family pedigrees, as for humans. All right, so two problems are presented below. In each, you're given a wild population. This simply means what is occurring in nature. The phenotypic characteristics of a wild population of fruit flies that were trapped randomly on a remote island. So in taking this sample, we're making some assumptions about this sample being representative of the total population. And then B, you're given cross 1 and 2, etc. The phenotypic characteristics of offspring from a controlled cross. The phenotype is the thing that you can observe. It is what is genetically expressed, and we have to make inferences based on what is expressed. So the phenotypes of the parents are indicated after each cross. Example, cross 1 male ambler crossed with female wild type. All right. So for each of the problems, analyze the results in each cross and answer the questions below. So problem 1, we're given a wild population of males and females along with their totals. We're given the wild type, oh, ambler, and total. So what does cross 1 tell you about dominance versus recessiveness of the alleles? Kind of wanting to understand a little bit better what this. Okay, so looking at these numbers, we see a pretty much even split of males to females, which is expected. We also see a fairly even split in amblers. In cross 1, we've got exactly even split for the wild type and not quite an even split, but close of the ambler. So because we get equal numbers of both phenotypes, it's it's really impossible to determine if one is dominant over the other. Since both phenotypes are approximately equal, we cannot determine dominance. What does cross 1 tell you about placement of the alleles on autosomes versus sex chromosomes? So this one we can talk about a little bit. Because the number of males and females in each phenotype is approximately the same, it's actually, again, it's impossible to determine if the allele is an autosome or a sex chromosome. I'm sorry, I misspoke there. So because number of males and females is approximately the same, we cannot determine if it's sex linked. So what to do about this? Well, that was our first cross. The real like magic in this experiment is the second cross starts to reveal um, what's hidden here. So in the second cross, we have zero males with the wild type and 0 females with the ambler type. So because all of the males are ambler, all of the males are ambler, and all of the females are wild type, this indicates that ambler must be recessive. And we know that it's recessive because it's only showing up on those males, and therefore it must also be sex-linked. So since all males are ambler and all females are wild type, ambler must be recessive and sex-linked. What does cross-2 tell you about placement of the alleles on autosomes versus sex chromosomes? And in your answer, show the chromosomal genotypes for the parents in the cross. I'm looking at the right thing here. Yeah, okay. Yeah. This tells us the alleles are on the X chromosome. So one of the parents must have been the Ambler female, and the other one had to be this wild type male. Yeah, okay. Moving on to the next problem. So we have our initial sample, cross 1. We're looking at monocle, bifocal, trifocal, spinner, or shiny. Okay, males and females. We have an uneven distribution for monocle and for spinner. It looks, well, okay. How do I want to make sense of this? Probably by color coding it. Let's see here. Ooh, interesting. Okay, so in our wild population, we have this kind of distribution here where we have some monocles, some bifocal, that BT gene, and some trifocal, a lot of trifocal with the spinner and fewer with the shiny traits. It's interesting. Okay, pay attention to that. But in our first cross, where we're crossing a bifocal spinner female times a monocle shiny male, we have absolutely zero monocles or bifocals. We have only trifocals. So what that tells me is that monocle and bifocal must be codominant, and the heterozygote creates some new trait, which is that trifocal. So monocle and bifocal are codominant with the heterozygote producing trifocal. Okay. Yeah. Sorry, took me a minute to get there, but yeah, trifocal is the emergent heterozygote being expressed. So when we cross that um, bifocal spinner female with a monocle shiny male, we only have the possibility of MT crossed with BT. And since neither of those appear, we know that there's a 100% chance that they're all going to be heterozygotes. None of those individual traits appear. It's all trifocal. Trifocal must be an emergent property, the phenotype of the heterozygous genotype. That's kind of cool. Neato. Um, so from these results, we can't really make an inference about spinner versus shiny here, right? Since they appear in equal number. So we won't. What does cross one tell you about the placement of the alleles on autosomes versus sex chromosomes? In your answer, show the chromosomal genotypes for the parents in this cross. So let's see. Because all the males and females are trifocal codominants, the the monocle and the bifocal alleles must be autosomal. So because we have approximately the same number of male and female trifocals, which are codominants, monocle and bifocal alleles must be autosomal and not sex-linked. And again, it's unclear whether the spinner versus shiny alleles are autosomal versus sex-linked because the numbers of males and females with each trait are similar. So you could get the same results if the genes were either sex-linked or autosomal. Come on. There. Okay. Same deal, but with another cross. Cross 2, we've got a monocle spinner female and a trifocal spinner male. So we know that that trifocal spinner male is heterozygous for the monocle and bifocal. And we don't know anything yet about the spinner. So the fact that they both have that trait will tell us, hopefully, should tell us something. And looking at this, we see that we do get some shiny. Interesting. But it looks like we only get shiny males, and we don't get any. We get absolutely zero, zilch, no shiny females. So that should tell us something about its placement on the chromosome. So what does cross 2 tell you about dominance versus recessiveness of the alleles? So this confirms that the alleles for monocle confirms codominance, basically. Come on. Codominance of monocle and bifocal. Yeah. Now, because we get some shiny and some spinner in this cross, um, even though the parents were both spinners, then shiny must be recessive. So because some offspring are shiny, shiny must be recessive, and both parents had to be heterozygotes or carriers of that gene. So this indicates, this indicates that the spinner and shiny alleles must be on the X chromosome. Oh, no, sorry, I got ahead of myself. It wasn't that both parents were heterozygous. I forgot about the sex-linked distribution here. It's not that they were both heterozygous, it's that it's X-linked because only the males express that. Shiny must be recessive and sex-linked, since it's only expressed in male offspring. Wow, even I can't read my handwriting sometimes. Cool. So therefore, I'm going to scroll off. I'm not going to scroll off to the side here. What's going on? Trying to give myself some more space here. And I killed it. All right. Just thinking about it. No? Nothing? Hmm. Technology's great when it works. At any rate, this would mean that the female parent was heterozygous for the shiny versus spinner gene, and that the no, yes, yes, the parent female was heterozygous, and the parent male had the spinner gene, which is dominant, and so the offspring, therefore, um, you wound up with 25% homozygous spinner, so dominant females, heterozygous spinner females, 1/4 spinner males, and 1/4 shiny males. You should see no shiny females, and the ratios of the others should be 2 spinner females, 1 spinner male, 1 shiny male. What does cross 2 Yeah, what does cross 2 tell you about the placement of the alleles? That's exactly what we were just talking about. Please come back. Oh, okay. Well, hmm. Let's move on, I guess. Analyze the pedigree and answer the following questions. The diagram below shows a pedigree of 3 generations in a family. Black circles and squares indicate persons with a genetic disorder. A square indicates a male and a circle indicates a female. The 2 males in generation 1 are siblings. The 2 males in generation 1 are siblings. Okay, so the 2 squares. One of them has the genetic disorder and one does not. Okay, so I think I really want my pen to work. It really doesn't want to. Hmm. There we go. I don't know what I did. Cool. So they're brothers. They have parents in common. And we don't know anything about them yet. So I'm just going to leave that bracket there to kind of help us. Our first question, looking only at the generation 2 offspring, generation 2, what can you say about the genes controlling the genetic disorder? Is the disorder caused by a gene that is dominant or recessive, autosomal, or sex-linked? So looking at this, if we look at A, the left side of this pedigree, we have a father with this genetic disorder, and we have one daughter who also has this genetic disorder. Interesting. We have a son who does not have this genetic disorder. On side B, father does not, but mom does. We have a daughter with and a daughter without, and a son with the genetic disorder. So the 2 males in generation 1 are siblings. Okay, so from this, um, this gene is most likely dominant, just based purely on how often it's occurring. If it's dominant, the gene may be either autosomal or sex-linked. Based on the data alone, there's a good chance that the gene, um, there is a chance that the gene is recessive, but for that to be true, the 2 brothers would both have to mate with heterozygous females in order to produce offspring in generation 2 with the disorder. So this is much less likely to be a possibility, though it is a possibility. So most likely, let's go ahead and write this. Gene is likely dominant. Okay. What additional information do you gain from examining the generation 3 offspring? So, looking at generation 3 now, the mating between 2 affected individuals is interesting. So lineage A, generation 2, we have 2 affected individuals here. Um, if the disorder were caused by an autosomal recessive gene, all of the offspring, 100% of the offspring in this cross would be homozygous recessive and have this disorder. But because we have one male who does not, because we have one male who doesn't have it, the disorder must be caused by a dominant allele. because gen 3 of lineage A is not 100% for the disorder, it must be dominant. Yeah, it must be dominant because if it was recessive, it would require, if it was recessive and both of the parents were expressing it, 100% of the offspring would be homozygous recessive, and they are not. What else can we tell from this? Okay, so looking at the information from generations 2 and 3, it's also likely that the allele is also sex-linked, since all daughters of affected males have the disorder, and males only have the disorder if their mother had it. So males only appear to have it if their mother had it. Excuse me. Yeah, that makes sense. All the daughters of affected males have the disorder, and males only have the disorder if their mother had it. However, given that this is a relatively small population, there's still a possibility that the disorder is autosomal dominant. So it's likely since all daughters of affected males have the disorder, and males males only have the disorder if the mother had it. Still a possibility. It is autosomal recessive. Or excuse me, autosomal dominant. Cool. We are out of time for this week, but if you want to stick around for a few more moments, I can kind of talk about this. How can the mode of inheritance be determined experimentally? So outline the experimental crosses you would need to make to solve each problem. 3 new traits have been discovered in a population of flies. Tapping, a behavioral mutant in which the fly taps one foot constantly, single stripe, and angular. The positions of the 3 genes on the chromosomes are unknown. Given 2 pure breeding homozygous lines and using an initial cross of normal, normal, normal females with tapping, single stripe, and angular males, describe the appropriate genetic experiment needed to establish whether any of these traits are caused by genes that are, for example, autosomal or sex-linked. So if we're testing to see whether this is autosomal or sex-linked, first we would just mate normal, normal, normal homozygous females. So let's do mate uh, norm, norm, norm females with tapping, come on now, single stripe, angular males. The phenotypes of the F1 generation, so that first cross, um, the F1 individuals will indicate which alleles are dominant. Next, mate the F1 males. Excuse me. Yes. No. The phenotypes of the F1 individuals will indicate which alleles are dominant. So F1 phenotypes indicate dominance. That's not gendered. I apologize. Um, then mate the F1 males with females that are homozygous recessive. Come on now. Mate F1 males times homozygous recessive females. Um, and made F1 females with homozygous recessive males. Once we do this cross with homozygous recessive males and females, we would examine the ratio of phenotypes for each trait in the offspring as a whole and compare that ratio for the offspring as a whole with the ratio for each sex. Let's see, control phenotype ratios to offspring as a whole and as ratio by sex. For example, if we assume normal is dominant in all cases, then the crosses would look like this. And then all of our F1 generation would be heterozygous for all three traits. And then in our next cross, so we're crossing this with, yeah, I do want to draw this. Our next cross would look like this. Come on now. Really? Well, you guys have done a Punnett square, so you should be able to finish this part now. But you would get some distribution. Um, to determine if it's linked on the same chromosome or unlinked, if the genes are not linked, we would expect the probability of offspring with a given set of phenotypes, for example, normal, one-stripe, angular, to be equal to the product of the individual probabilities for each occurring in separate crosses. So in other words, the chromosomes segregate. This law of segregation and independent assortment. So to be equal to the product of individual probabilities for each occurring as separate crosses. For example, if the genes are autosomal, then the F1, that first cross mating, is heterozygous for all three traits times homozygous recessive. And if the genes are not linked, we expect to see 50% normal, 50% one stripe, and 50% angular among the offspring. And the probability of all these characteristics showing in the same offspring is 50% times 50% times 50% equals 1/8. Um, so if A and B are linked, we would get different results, right? If the first cross becomes big A, big B slash a, little b times a, little b slash a, little b, um, you'd get 50%. Well, how do I? Yeah, the technology breaking down is really cramping my style here, and we're over time. So I am going to give you the rest of your evening. Thank you for coming. Don't forget to submit your worksheets for that sweet extra credit. I apologize for the technical difficulties. I will try and have this resolved by next week. There is no live stream next week. I will be recording something probably over the weekend, and that will be available on the PSU Learning Center YouTube channel. And again, if I can't get the technological difficulties figured out, then we'll just have to figure something else out. But that's all I've got for you this week. Thank you for your time, and hope you enjoy the holiday next week.